Hello everyone, welcome to the ITLS Academy. Today we are going to perform another experiment in the series of microbiology techniques. So today the experiment that we are going to perform is a effect of temperature on the growth of microorganisms. Okay, so for this experiment what we have to do, first of all we prepare a agar plate. This is our agar plate. See how we prepare it. First of all, we prepare a nutrient agar media with a composition for a 250 ml. For 250 ml, we required agar 4 gram, yeast extract 1.25 gram, and then your NaCl 1.25 gram and peptone 0.75 gram. Okay, then after that we mix all these ingredients into a 250 ml of the distilled water. Like we mix agar separately into 100 ml of the distilled water, and in 150 ml of distilled water we mix yeast extract, sodium chloride, and your peptone. Then after that we prepare our uh, we maintain the pH and after that we sterile it into a in a autoclave at 121 degrees Celsius temperature and 15 psi pressure, and then for 20 minutes. After 20 minutes, what we have to do, we take out the media from a autoclave and we pour it into the plate and leave it for a solidification. Okay, see, this is solidify. Now at that time what we have to do, we perform a serial dilution. This is our serial dilution tubes. Now how we prepare the media, how we sterilize the media, how we do a serial dilution, and how we maintain the pH. These videos are already uploaded in our YouTube channel. You can visit our YouTube channel and see the video related to these topics. Then you can better understand how we perform all these experiments. Now after that, the experiment that we are today that we are going to perform is what? It is a effect of the temperature. We all know that bacteria required a optimum temperature for their growth. Okay, if there is a slightly change in their environment, then it directly affected the microbes. Okay, and also the growth of the bacteria are affected by the five factor, that is your oxygen, carbon dioxide, temperature, pH, and osmotic pressure. Okay, so the first factor that we are going to discuss is that the effect of the temperature, and the optimum temperature are required for the growth of this bacteria. Okay, on the basis of the temperature, uh, these bacteria are divided into a three category. The first category is your psychrophils. Psychrophils are what? Which can grow at a low temperature, and the temperature ranges between 0 to 20 degree Celsius. And the example is your ETC. Then the second type of the these bacteria are what? That is your mesophilic, which can grow at the temperature range between 20 to 40 degree Celsius. For example, lactobacillus. Most of the pathogenic microorganisms can grow between these temperature. Now, what is this pathogenic bacteria? The pathogenic bacteria are those bacteria which have a ability to cause a disease into a human being. Okay. Now, the last category is your thermophilic bacteria. Thermophilic bacteria are what? Those bacteria which are what? Which are heat tolerant, means that they can grow at very high temperature, like a temperature range between 50 to 80 degree Celsius. For example, Thermus aquaticus. Okay, so these are what? These are the ranges of the bacteria. Now today what we have to do, we have a 3 Petri plates, okay, in which we are already pour the nutrient agar media. Now in first plate we put A, and also we write down a storage temperature, that is 5 degrees Celsius. See, A plate, storage temperature 5 degrees Celsius. Now second plate is your B temp, and storage temperature is what? 28 degrees Celsius. That is your today's room temperature. Okay, so you have to like mention the second plate according to your room temperature. Today our room temperature is about 28 degrees Celsius, so we write down 28 degrees Celsius. Now the third plate is C, storage temperature 37 degrees Celsius. Now what we have to do, we take this from a serial diluted tube, we take a solution, or you can say that we take a microbial culture, microorganisms, and then uh, we inoculate this into a Petri plate. After inoculation, we put that Petri plate according to the temperature. Like in a first Petri plate, we write down a 5 degrees Celsius. So after inoculation, we put that Petri plate into a 5 degrees Celsius for a 48 hours. Now in a second tube, I write down a room temperature, that is 28 degrees Celsius. So at this plate should be kept at a room temperature for a 48 hours. Now in a third test tube, in a third Petri plate, what we have write down? We write down a 37 degrees Celsius. So after inoculation, what we have to do? We put that Petri plate at inside the incubator for a 48 hours. Then after 48 hours, we see result all in all these plate. Now if we talk about the first plate, then it means that this first plate which put into a 5 degrees Celsius, which means that there is no growth of the microorganism take place, because we kept it into a into a 5 degrees Celsius, that is your refrigerated condition. Okay, that's why it does not show any growth. Now at 28 degrees Celsius, that is our room temperature, there is a slightly growth of microorganisms we observe, but at 37 degrees Celsius, there is a maximum growth that we observe, which concluded that the bacteria can grow at an optimum temperature, and most of the bacteria can grow at this temperature. So at the end of the video, I will attach the photo related to this experiment, like the image which are related to this storage at 5 degrees Celsius, then storage image related to 28 degrees Celsius or room temperature, and storage at 37 degrees Celsius. Okay, now first of all, what we have to do, we have to inoculate this. Now we take a 10 to the power 3 dilution, put it into a first Petri plate. We take a same dilution for each Petri plate. Then inoculate at a second test tube. And inoculate into a third test tube. We perform a spread plate method. For this, you can also use a spreader. Okay, how we do a spread plate technique, the video related to this are already uploaded. You can visit our YouTube channel and see. Now what we have to do, we kept these petri plate at their temperature. Like the first petri plate should be kept at a 0 to sorry at a 5 degrees Celsius, and then second test tube kept at a room temperature, that is your 28 degrees Celsius, and the third test tube kept at a 37 degrees Celsius. Now at the end of the video, I will attach the image uh, related to these experiment, which show you that the first petri plate which kept at 5 degrees Celsius, which it does not there is no, it does not observe any growth, but in a B test tube, which in a B petri plate that we kept at a room temperature, there is a few microorganisms growth are observed. But in a third petri plate which kept in a which kept in a 37 degrees Celsius, it show a maximum growth. So this is all about the effect of the temperature on a bacterial growth. I hope you will understand. But if you have any query related to this experiment, you are freely to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms. That is your Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn. And subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.